during the morning. Okay, so it's my pleasure to have Ava Khamseh.
It's actually Khamseh.
Khamseh, sorry, Khamseh. I've messed up 2 names. From Edinburgh, and she's a lecturer, and she's actually joined between informatics in Edinburgh and the Institute for Genomics and Cancer. So she works jointly between the medical people and the informatics people, which is a great combination. And she's going to talk about higher order expression dependencies. And I was trying to make the morning a little bit thematically linked, so I was trying to claim that this is related to networks, but I'm not
sort of.
Sort of.
Okay, thank you very much for the invitation. I've been told it's 50 minutes plus 10 minutes of questions, but actually I prefer if you guys ask questions in the middle.
But don't worry, they always do.
Yeah, okay, perfect. Yeah, so please just shout out and we can discuss. Okay, so first of all, I just want to thank all my collaborators on this project that lasted multiple years. So they span genetics and cancer, School of Physics, and School of Mathematics. And this has been a cross-disciplinary journey where we started in 2017 and we ended in 2025 with 2 pieces of theoretical work that I'll try to summarize, and then finally with a major application to single-cell RNA-seq data to infer cell states. Now, our study really started by looking at the word interaction, so you'll hear that a lot during this talk. And so this word appears elsewhere as well. I'm sure you've heard it this week. It can be a bit vague, so I want to say that this what we would need to do is to think about a relevant question on the biomedical side and need a context, and that context changes the interpretation of what this word might mean. On the physics and mathematics side, we wanted to have a robust quantification of interactions in a well-defined mathematical manner. And this was a bridge that we had to make between the 2 sides to make sure the thing that we build on this side actually has a use on the other side of the bridge. So our aim on the physics and mathematics side was to quantify dependence amongst large numbers of variables beyond pairwise. And you'll see in this application that we're looking at gene expression data at single cell level. And on the biomedical side, the aim ended up being actually determining types of cells and their states in order to be able to compare at high resolution disease progression as compared to normal healthy tissues. So the central dogma in biology starts by DNA makes RNA, makes protein, very rough. Now, DNA in all of our cells is identical. To first approximation is identical. Given a person, all their DNA in their cells is the same. Now, what makes a liver tissue, and a heart tissue, and a brain tissue, is how different bits of DNA is expressed in terms of RNA counts. So we have 20,000 protein coding genes in the human body, but given a particular tissue, maybe around 10, 15,000 or so order of magnitude, different genes are expressed. And this whole thing is only 1.5% of the whole DNA. And then from RNA, we make proteins, and actually a lot happens in this step. A lot of regulation happens in this step, but for some nice, interesting reasons, RNA seems to be a good approximation for what happens. So people for years have been now looking at RNA expression to study disease progression, to then inhibit certain pathways to find therapeutics and so on. And so despite a lot happening in this step, it seems that it's a good approximation to look at the step in the middle. So what we're dealing with is a large number of dependent variables when we start looking at RNA counts. And as I said, sort of given a particular tissue of interest, everything in biology is relative. So we want to be comparing healthy versus a particular disease state. And we want to see how these cells are changing in terms of their expression. And based on those differences, try to figure out, well, first detect the disease, detect the right cell type, detect the right cell state, and prevent progression, or in fact prevent the disease. That's the general aim here. For those of you who haven't worked with this kind of data, basically you take a piece of tissue, maybe from a human, perhaps post-surgery, or an animal model of the particular disease. They take the cells from the tissue, dissociate them into single cells, and from each cell, we extract RNA counts. So we literally just count how many times this kind of gene is expressed versus another gene versus other gene and so on. And this is done through sequencing. And long story short, the data looks like a cell by gene expression count matrix. And in what follows, I'll end up binarizing it as 0 is not expressed and 1 and above for anything that is expressed. But then we'll come back to the full count data later on. So the context we are working with is molecular biology. The data is single cell RNA sequencing, so that count matrix of cell by gene expression. And it turns out that the interpretation of gene expression interactions is not trivial. So again, I'm going to come back to what it means to say gene expression from data is interacting. So the study of interactions come up in very many different areas of science. So my background is actually in theoretical particle physics, where we're trying to study interactions amongst fundamental particles. Then through these physical laws forms everything we see in the universe. We have similar kind of thing in statistical physics. Perhaps we're looking at magnets on a lattice, and magnets that are proximal are interacting in a certain way. We'll define, again, described by an energy function. Folk in machine learning sometimes talk of interactions amongst the nodes in a neural network. And again, I want to now distinguish in the first step the difference between a dynamical or a physical interaction and one that is statistical or probabilistic. And what we'll end up seeing later today is the object we have quantified for the purpose of biology is of that probabilistic statistical nature and not one that is related to a physical interaction. But mathematically, they may at times be written down in very similar ways. Okay, so we're now going to start on one side of the bridge and build the mathematics, and then at some point we're going to meet the other side, and we hope that we actually land not off from each other on this bridge. So there is a problem in physics known as the inverse forward and inverse problem. The forward problem has the goal to provide a macroscopic description of nature by observing observable quantities, by deriving observable quantities from underlying laws. So here you obtain observables given some energy function and its parameters. Now, the inverse problem to this is you start with observational data instead. You don't know what those parameters are, and you actually want to infer microscopic properties by just looking at the data. So this really becomes a statistical probabilistic problem, whereas the first one really starts from certain laws of nature, and then we build up from there. So what we're interested in this case is the inverse problem. So we want to look at some data and from that extract dependencies amongst the various variables. So there is this famous model in statistical physics known as the Ising model. And you can imagine it as a two-dimensional lattice where there is a magnet at every node on the lattice. Now, the magnets can either point north or south, up and down. And these are represented by this sort of SI and SJ. And there is a coupling or interaction between these if they are nearest neighbors, and that is represented by Jij. So here we have in probability distribution, the normalization. So this Z is the partition function, and we have the energy function here at the top. And you see here that this energy function is entirely described by nearest neighbor pairs with a strength of coupling amongst them. And this is a well understood model by Onsager in 1944. And it's possible to actually try to generate lots of data from this system. And the reason we wanted to start with this system is because it's well understood, and every theory we develop should, at the very least, be able to extract this Jijs from the data. So going back to the inverse problem, we want to generate data from the system. We blind ourselves to what that coupling is. And we want to come up with a technique where we can figure out what that coupling is. And because we know the ground truth, at the end, we can compare and see if we've gotten the right answer or not. Now, you can imagine that if you are in a very cold system, these magnets are either all pointing upwards or downwards, very sort of contracted in a way. But when the system is very hot, they're all very wobbly, and the interaction between them, or this Jij, is very small, and then they are just sort of rotating in different directions, up or down. So this is going to be the data set that we will use for our simulation. Now, separate from this, we need to actually solve the Ising problem. So we have to be able to estimate those interactions from data. And we started this journey by looking at restricted Boltzmann machines, or RBMs, to address this problem. Just want to note that RBMs, so at least Boltzmann machines, were explicitly mentioned. They were developed by Hinton based on part of Hopfield's work, and they won the Nobel Prize in 2024 on this type of foundational neural net algorithm. So they have a very simple structure in a way. There is one visible layer and one hidden layer, and that's it. The word restricted applies to the fact that the visibles amongst themselves don't have any connection. So you see no line amongst the visibles. And similarly for the hiddens, but every visible is connected to every other visible through some hidden connection. It has a very similar shape in its energy function. So again, it's a normalized probability here. You see the energy function is a function of the visible and hidden, as it should be. And then at the top, we have an interaction, if you like, between each hidden and each visible, represented by these lines. Then we have a bias terms for the hidden variables in C. And with B, we denote the bias terms for the visible units here. And the way to train these machines is by taking the empirical distribution of the data and minimizing that difference with the distribution of the RBM in terms of the parameters of the RBM. And this is done through a KL divergence loss function. So if you expand this quantity, you'll have a bit that entirely depends on data. So there's nothing to be done there because you cannot minimize anything there. That's just the empirical distribution. And the second bit has a dependence on the parameters of the machine. So effectively, what you're doing is you're tuning these Wij's, Ci's, and Bi's in such a way that eventually the machine recovers the distribution, the empirical distribution of the actual data. And this kind of model is a generative model. So once you have fitted W, C's, and B's well, you can generate data from the machine that mimics the data you put into the machine. Okay, so what we're going to do first is we're going to train this machine on these Ising configurations, so these magnets, and then later on we're going to do this for single-cell RNA-seq. So let me show you an example of a one-dimensional Ising model. So rather than having 2 dimensions, you can actually analytically solve the system of magnets if they're just in a line next to each other. This is for a system with 6 variables. So there are 2 to the 6 possibilities, and they are depicted on the x-axis here. And on the y-axis, we have the probabilities, and of course, they sum to 1. So what you see here in the dashed line is the actual probability. This is analytically known, so there's no empirical trickery here. This is sort of closed form. We know exactly how it looks like. So it's a great test case for us to understand if these machines are able to learn this kind of probability distribution. And what you see here in purple is that the machine starts by a random initializations of these parameters. And as the machine is training through some procedure by minimizing this KL divergence, you see that it starts getting into these dips. So as the colors are changing towards green and red is when we declare the machine as trained. It's mimicking this landscape of the probability distribution pretty well to a large degree. Okay, so brilliant, it works in this case. But how do we, generally speaking, when we don't know how the probability distribution looks like, tell if we have trained well, beyond the usual sort of loss function and so on? So one way of doing this with a generative model is to compare the moments the machine is generating with the moments of probability distribution from the data. So what are the moments? Averages or variances, and then again, sort of skews and so on. So you can try to go higher and higher in the moments of the data. So as an example, we can essentially take the average of these magnets being up and down. We can estimate that from data. We can estimate that from what the machine is outputting. we can quantify their uncertainty and see if we're agreeing with what's being generated. And any probability distribution can be written as an expansion in the moments. So the higher up you go in the moments and you check that everything is being learned, the more sure you can be that you're close to the actual probability distribution. So these are just a bunch of examples of quantities, observable quantities we can estimate from data and the model. So here on the x-axis, you see as the training is progressing, and the actual value is in these dotted black lines, and the machine is in colored values, again, as it's training, and we've done this for 3 different temperatures. So 1.8 is cold, 3 is hot, and 2.2 is near what we call the critical temperature. And what we see here is that the first moments, like magnetization and energy, actually you learn them pretty fast. So it just starts maybe a bit off, but very quickly converges to the actual value from the data. And then the second moments are harder to learn, and perhaps this is completely expected. So we see that, especially for this one in the middle, it starts way off, wild fluctuations, and then at some point, again, it converges to the true value. Now, the good news about this is that it doesn't matter what your original data is. If you don't know anything about the true parameters that generated that probability distribution, you can always measure empirically the moments from the data and compare with what the machine is generating. And it turns out the higher up you go in the moments, the more difficult it becomes for the machine to train. Again, partly expected. But if you really want to measure something precisely, it's not good enough. And that's what we showed in this work, to just look at the loss function. So if you are working with a generative model, whether it's RBM or something else, it's always good practice to look at these higher order moments to see how the machine is actually behaving. So we wanted to go sort of beyond training criteria and actually estimate interactions amongst the visible nodes, if you like, or our spins or our magnets. Now, it turns out something curious happens with the energy functions of these machines. And that is, you can take the energy function, the marginalized one, where you've kicked out the hidden variables, sum them up. It turns out that if you use a sort of moment-generating expansion, you have something, let's not worry about the coefficient, it's something complicated in terms of the parameters of the machine, but it's something times Vj, where V is the visible unit, something times Vj times Vk, then 3 of them, then 4 of them, and so on. Now, when you look at this, and you can again do this for any of these energy functions, you see that this is an asymptotic expansion. On face value, it looks like an infinite series. However, if you're dealing with binary data in the 0-1 basis, 0 and 1 have the special property that 0 to the power of n is 0, and 1 to the power of n is 1. So actually, the series can be resummed. So I'm not going to show you, this is like 3 or 4 pages of algebra, but if you resum the series, you can actually figure out what is a 2-point interaction between 2 variables, Vj and Vk. So all these coefficients come together into one big expression, and it turns out that it looks like this. So the crucial thing about this is that this is entirely closed form. So no matter what the data is from this machine, you can extract this interaction between the variables. And in particular, you see that it's a function of the connections between the visible and the hidden, and the visible biases, and the hidden biases don't in fact appear as part of this expression. And then you can generalize this to higher orders. So this is for a 3-point interaction. You can do it for 4-point interactions. You generalize it to endpoints. Yes. So it's the coefficient that sits behind Vj, Vk. So V were the variables. So it's a coefficient that says, okay, so with this strength, Vj and Vk are related. In the Ising model case, it would be these Jijs. What I'm showing here is a 2-point interaction of the Hamiltonian of this machine, of the energy function of this machine. Now, if we're training everything correctly, this guy here should be equal to the Jij in the Ising because it's learning those couplings. Okay, so that's what we get. So on the left-hand side, we have the ground truth that, well, okay, we know what the ground truth is, but the machine is blind to that ground truth. And on the right-hand side, we have the estimate from the RBM. And I hope you can appreciate that the exact patterns are extracted and in a data-driven way, and it exactly matches. The color you can ignore because depending on the temperature, this changes. But the structure is basically identical, and in value, they're identical depending on what temperature you're going to look for. And the exact patterning here is related to the fact that nearest neighbors interact. If magnets are not nearest neighbors, the interaction is 0, so that's the black bit here. And these other patterns are related to periodic boundary conditions around the lattice. This is 2D.
This is 2D.
Yeah.
So is it a 2D spin glass?
It's easier than spin glass. So in fact, all the nearest neighbors have the same coupling in this particular case.
Phases.
Yeah, so before we even go to the before we even go to the phases, so one of the things we wanted to look at is also like what happens at critical temperature, if it's more difficult to learn. Not really. So at least in a finite lattice, they all seem the same. So one other thing we can do is to actually look at how the couplings are being learned as we increase the epochs, as the machine is training. So at the beginning, when the parameters are initialized randomly, there is nothing, there's no pattern. And then as the machine is learning, you sort of see that this nearest neighbor next to nearest neighbor pattern appears. And then, more and more, these kind of spurious interactions go away. And at the end, when we declare it trained based on the criteria I just defined, then you have a very clean pattern. So at this point, we're hailing victory because it's like, well, yeah, it learns from data and we recover the ground truth. Yes.
If the J's were random,
yeah,
plus minus, there's 2D, isn't it can be hard to find the ground state when I think spin glass in 2D?
So we haven't tried it on spin glass. I'll have to think more about that. I don't think there is any particular reason why it wouldn't be able to do that.
I think the ground hard like finding.
I think there are people. Yeah.
Energy about state. So the problem. So I think it's got spin glass.
So I think there is actually a paper that's I think applied RBMs in the spin glass case, but I'll have to refer back to that. I don't remember. I'll have to look that up. But yeah, in our case, it's nice and symmetric. Yes, it's nice and symmetric, and it's just So the other thing is actually the number of training examples. If you start with only 200 training examples, it's terrible at training. 2,000 training examples, it's bad still, right? You see that there's a lot of these spurious interactions there. And even at 10,000 examples, it's not doing pretty well. Because we want all these bits to be entirely black, and they're not. So they should be 0 because they're non-nearest neighbors, and they're not. So the training I'm showing here is based on 100,000 training examples. So it's very data hungry. So with this in mind, we had learned a couple of things. So we understood well the training criteria for the RBMs in terms of log likelihood, loss, free energy, reconstruction error, and the moments generated. And again, I want to emphasize the importance of this for generative models. They're successful at estimating high order interactions in a given system of binary variables with the caveats that was just mentioned. And generally speaking, they need a lot of training examples. Now, the problem is that we still need to deal with potentially very large number of variables. So the 2D system I showed you here is an 8x8 lattice. So that's 64 variables only. And when we're dealing with G networks, we're looking at many more. Right? So even if you only take into account the highly variable genes, you're looking at thousands. And they're not very convenient to train, or at least they weren't in 2018. I don't know many, not aware of the latest developments on training, but at the time it was quite annoying in terms of time and hyperparameter tuning. Okay, so we realized that actually on the maths physics side, we had to do more here. And so at this point, it was 2020, I was attending an Alan Turing masterclass by Mark van der Laan. And he's the pioneer of this statistical approach called targeted learning. And the general idea is that ask the question that you're actually after, and don't worry about fitting something more complicated. and make sure that the quantity you end up estimating has the least bias possible with the best power. It sort of makes sense when we think about biology. So the question I had here is, why on earth am I bothering fitting the full joint probability distribution when what I actually need is just the interactions, 2 point, 3 point, 4 point, and so on. So those of you who may have come across targeted maximum likelihood estimation, we're not going to go through that because I'll end up estimating this interactions not by fitting, but by counting. If that doesn't mean anything to you, don't worry about it. But the whole point of this is to define the target statistical quantity of interest, not as a property of a parametric statistical model, not as a coefficient in this particular model, but entirely model independently. Be clear about the question you're asking, come up with a good definition that you think is a good definition in this model independent manner, and then just go ahead and estimate that quantity that you actually care about, and don't worry about everything else. So that's the general idea here. So how did we come with this? So there's no theory in biology, unfortunately. Yes?
Could you expand on that a little bit? I didn't quite get.
So, okay, so obviously, a restricted Boltzmann machine is a highly parametric model with all these weights and so on. And what RBMs do is to fit the full joint distribution. And then from that, we're extracting the coefficients. The idea behind targeted learning, and I really encourage you guys to look into this, is that even if you're fitting something more complicated, typically you're interested in a very specific question. When you speak to biologists or biomedical researchers, rarely ever do they care about what the joint distribution is. Right? They want to know, I don't know, some expectation value or conditional expectation value. They want to know if a person under this treatment versus that treatment or in the population is better or worse. And often these questions can be written down as difference of expectation values or some change in that. And that you don't actually need to, in order to define it, you don't need to refer to a particular model or particular coefficients in a model. So the idea is to say, start with a question, start with a target, and that's where the name comes from, and actually try to do the minimal amount of work to, with a maximal power and minimal bias, estimate that quantity. Right? So at the moment, I just want to define what an interaction means to me, and then worry about fitting later. Okay. So when you do a fit, because at some points, especially when you have continuous data, I have binary data, I don't have that problem. But when you have continuous data, you do have to end up fitting. And then there is a machinery called targeted maximum likelihood estimation that then targets your question, your quantity of interest, and guarantees in a mathematical way that you set the bias to 0. So again, this is very important for real-world evidence studies, if you come across that. So, okay, so the question was, what is an interaction to us? So we wanted to come up with a sensible model-independent statistical definition. And again, model-independent here means that I don't want to be looking at a specific model or its coefficients. So if you just ignore this conditional here for the time being, doesn't really matter. So for one spin, or gene later on, as it turns on versus off, what this quantity is measuring is essentially a log odds ratio. What is the likelihood of spin I being 1 versus 0? Or what is the likelihood of a particular gene being expressed versus not? So for 2 spins, we can generalize this quantity in the following way. If you look at the first one, it's still switching from 1 to 0, whilst the second one is set to 1. And then the same thing, except here, the second one is set to 0. So we're still switching the gene on versus off, or the magnet up versus down. But in the first term, the second is set to 1. In the second gene or spin is set to 0. So this is an odds ratio of spin I with spin j being 1. This is the same quantity with spin j being 0. And the interpretation of this quantity is, as a generalized log odds ratio, is does the likelihood of spin I being 1 increases or decreases depending on the status of spin j? So if the two are completely independent from another, this quantity will be 0. If there's a dependence, you end up seeing, and then you have to, of course, statistically quantify with error bars at finite sample, if that is actually significant to worry about. But you see, what you see here, there are no coefficients. We're just talking about probabilities and ratios of probability. So this definition is model independent. In our case, it's symmetric. It makes sense to us for a variety of reasons that I won't go into, but at least it's model independent. Okay, so as I said, when there's no interaction, at least this makes sense, and this is 0 when the two genes or spins are independent. Okay, so the task at hand at this point was to demonstrate if this definition, and we're dealing with binary data, if we can extract the Ising couplings just from this definition, one. And the second, if this is the right definition for interactions or a good definition for interactions, can we actually extract what an interaction is in an RBM? Because we already had it analytically through that big resummation. Can we extract the same quantity out? And it turns out the answer to that is yes. So I'm going back to the RBMs. We had this energy function. We had the connections between the visible and the hiddens, and then the biases in the hiddens and the visibles. We marginalize out H. We end up with this visible probability distribution. And I just told you that you have to do a painful asymptotic expansion and resummation. And if you do that, you get a closed form expression that looks like this. And the task here is to see if I can just take this quantity, plug this in, and get that within one line instead of pages of algebra. And it turns out the answer to that is yes. So through this formulation, we can actually do it together because it's very easy. So this quantity is telling us, put the first two to 1, then to 0, and then the cross term 0, 1, and 1, 0, and then set the rest to 0. So let's do that together. We have the first two variables saying being 1 and 1. So you see that as a result of that, you have a term here with a W, J, and then sort of a V1 that survives. And then there will be another one from V2 because it's a sum of all nodes J. And that's the two terms you see here. And then there will be this CI term, which is what appears here. And then the 1 term, which is here. This guy cancels between numerator and denominator because it's a ratio, so you don't need to worry about that. The partition function also cancels, so also brilliant. We don't have to deal with that. So this is the term, the 1, 1 term. And then similarly with the 0, 0 term, you see that all these V's go away. So the only thing you remain with is 1 plus each of the CIs. And then similarly for the other terms. And this quantity is identical to this up to a log of what we had derived before. So no asymptotic expansion and resummation required. And just by reading off the definition from the actual model, we can extract what the interaction is. So we were like, well, great. We can apply this to any energy-based model. And we did this for Ising. Turns out if you do this for Ising, you get exactly surprise, the JIJs in the Ising case. And here's the result and actual data. So again, analytically, we know this is going to work, but let's look at numbers. On the x-axis, we have the temperatures of this Ising model at different values. So again, from cold to hot. In orange is the actual known theoretical value, which is the ground truth following this pattern. And in blue, we see what we had with RBMs on 100,000 samples. And in red with 10K samples, only just counting. We're literally just counting because you see, this is binary. So you can just count how many times you see 1, 1, how many times you see zeros, how many times you see the cross terms, and then bootstrap to get your error out. So really simple. I mean, it's ridiculous, right? So we ended up getting these results. We're like, well, actually, so we don't actually need to train any machine. We just take the data, start counting, we bootstrap, and that's the answer. Yes.
What is condition of that?
Okay, sorry, good point. So this is conditional on the nearest neighbors. So Ising has a special case where you entirely essentially depend on your neighbors that are nearest to you, and there's no interaction with anything outside of that. So there's a caveat here that I'm actually inputting that information. So that's a very important question. So I'm inputting that I don't know what the value of interaction is, but I know that they only interact with things nearby. If you don't have that information, and this is referred to this thing here, so I'm being a bit cheeky, I'm inputting that actually these two, I and J, only depend on their nearest neighbors. If I don't have that, I have to infer that somehow. And we'll end up doing that actually in the gene expression case. Okay, so that's exactly an on-point question. You'll look at this and now you'll tell me, well, if this space is very large, you're computing a conditional that is very difficult. So say I'm dealing with 1,000 variables. I'm telling you I'll do 1, 1, and then I'm demanding the rest to be 0. I'll be in very bad shape very quickly with data. Now, as I said, it turns out that all you need to worry about is, in the Ising case, the nearest neighbors of these two. If you're dealing with a directed graph, you need to worry about, if you've come across that, a Markov blanket. So that's the minimal set of variables. As long as you condition on those variables, the first two become independent of the rest of the system. So actually, you don't need everything here. You need only the essential information in the conditional. And once you condition on that essential information, extra variables don't add any new information, and that can actually give you a lot of statistical power using this observation. Now, of course, now you have to go and estimate that from data. So the problem is a little bit shifted, but it turns out that actually in the causal discovery literature, there are algorithms, and we'll not go to that, that will be a separate talk, called Peter Clark and score-based algorithms that can infer these kinds of dependence graphs in a very efficient manner. Now, I want to emphasize, I will not be making any causal statements here, just using the algorithm as a means to an end to get at associations. And the reason behind this is a lot of these causal discovery algorithms have heroic assumptions when it comes to causal discovery. So for a general dependence structure graph inference, and there are many numerical tests we've done to demonstrate this, this is good enough, but I'm not going to end up telling you that this gene influences that other gene in a causal mechanistic manner. So I'm just going to not do that. Okay, so now we have something that looks like it works. So we want to apply this to real data. So just to remind you, we were in this situation where we had a cell by gene expression count matrix, and we want to compute dependencies at high orders amongst all the genes. Now, we don't have that nearest neighbor structure anymore, so we can't make those assumptions. And the number of variables is a lot more. We constrain on some top number of highly variable genes, in this case, 1,000. Many of the algorithms you work in this space actually do that. When you do dimensionality reduction and clustering, actually do that. And in our case, actually, there is minimal gain going higher, because the less genes vary, the more they're going to depend on other genes. So at some point you reach a plateau, and then adding more genes is actually just a computational pain. There's no point doing that. There's no gain. Okay. So the algorithm starts by assuming every gene is dependent on every other gene. And then through iterations of checking for dependence pairwise, it starts removing these lines. And then in the next level, it does conditional independence testing. Yes.
So just to clarify, what does it mean for two genes to be dependent?
So you have to come up with a sort of notion of dependence. So that kind of generalized log odds ratio is one of them. It could be as simple as correlation, for example. So obviously there's not, if you're dealing with continuous data, there are many notions of dependence, and a correlation is just one of them, let's say Pearson or Spearman, etc. For binary data, it's a lot more restrictive. So something as simple as a chi-square test you can do to get out this kind of thing. So round one, pairwise. Round two, conditional on one gene, are two other genes dependent or not? Now conditional on 2 genes, 3 genes, 4 genes. And then the algorithms keep running. If you do this naively, it's super exponential. But these algorithms that were developed by Peter Clark, and then the MCMC variants on top of that, have actually come up with very smart ways of doing this. Not super exponential. I actually don't know how it grows. It's not great, but it's far better than super exponential. I think it's polynomial, actually. And the higher up you go, the more sparse the graph becomes, the faster it will start learning. So it could be that for some tissues, this is really fast. And then if there is more connectivity across the genes in a different tissue, this is a bit more difficult. But with 1,000 variables, a couple of hours, it's not too bad. Okay, so now we have this kind of network structure, if you like, and I am very much suggestively not putting arrows on here because I don't want to make any causal statements. And so one of the nice features now is that anything that looks disjoint, at least at finite samples, so these could be housekeeping genes that don't really care about what's happening to other genes, they don't interact with anything, so we don't need to worry about those. Then we have other ones that could be hubs, so they have connections with many other genes. And so, for example, if I want the interaction between these two, I only need to worry about the status of these other two. And once I know something about that, the Markov blanket, I don't need to worry about the rest of the system. And maybe if I want the interaction between, I don't know, this guy and this guy, again, there is some nearest neighbor structure, if you like, that I worry about, and the rest is fine. Okay, so, and again, this is something that is generalized to endpoint interactions. In practice, we go up to 7 points, because every time you go higher in this order, the error bars grow, and at some point, you're basically just computing 0 within statistics. So there's not a lot of point to do that. So with this, we developed this algorithm called STATA. So here's where we are on the steps. We start with a cell by gene expression matrix. We infer this, I'm going to call pseudo-causal graph. So again, you don't need to worry about the arrows, but just in general, some kind of structure. The structure helps us increase power of estimation. So it's really a means to an end to compute these endpoint interactions. And now we have things that says these 2 genes are dependent, and then these group of 3 are dependent. That's group of 4, group of 5. So really doing something beyond pairwise. And the reason for that is we believe biology is not a pairwise dependence amongst variables, but it's actually something more complex. And we see that in data. You can quantify these quantities. Yeah.
Another clarifying question. Why is estimating these interactions useful from a biological perspective?
I'll get there. Yeah, it's a very good question. And it took us 2 years to solve that. So what is the biological interpretation of these endpoint interactions? And does it actually help answering molecular and cell biology questions? So it sort of required a lot of patience from all of us to get to this point. But yeah, is it useful? So this is the point where we have to start trying to understand what it means for gene expression to interact. So the first interpretation we thought of, and we thought that probably wouldn't fly, but let's analyze that, is that these model-free interactions represent physical interactions amongst the molecules within a cell. Now, there is literally absolutely no reason for this to be the case, because gene expression is not even protein, right? It's like gene gets expressed in a cell, stuff happens, stuff gets regulated, we'll end up with proteins. But we did check for this. So where we knew that certain genes later on form proteins that form complexes, we looked into that, and there was no evidence for this as expected. The second interpretation is that if we look at our network, there is a sense in which the head nodes are transcription factors that are regulating gene expression, and then the targets are targets of those transcription factors. And again, I told you that these causal discovery algorithms have heroic assumptions. If you're not dealing with perturbation data, you have to either make many assumptions or you can't really trust it. Nevertheless, you have some sort of structure, you have some sort of directionality. So we asked the question, is it the case that the parent nodes are enriched in regulators, transcription factors, and their children are enriched in the target genes of those transcription factors from known biology? It turns out that this is true. I mean, there is an extent in which you see an enrichment in this direction, but on a gene-by-gene basis, hand on heart, I cannot say, yes, this is a transcription factor and that's a target. So not confident enough, though, that there seems to be a general structure there. We thought, let's just leave that aside. What we ended up seeing is that these interactions or these dependent structures actually are pointing to cell types, cell subtypes, and cell states at very high resolution. And this turns out to be very helpful, that I'll try to show you. So long story short, these are not dynamical physical things. They are rather a statistical dependence across the genes that then can be used to construct or predict cell types and cell states at high resolution. So this is where we are. So we had these high order interactions. And of course, you can always define a null. And the null distribution in this case is if I have 3 genes, they have counts, but their counts are completely independent of each other. So you can think of it as a binomial coin flip, where the genes are not really, they don't really care. And what we want to know is, do we have sets of high order interaction that are more than expected by this chance of randomness, this null distribution? And it turns out you can see what pattern is driving these interactions. So if you have 3 genes, the possibilities are that the 3 are 0, one of them is 1 and the others are 0, There are pairs that are 1, and then all 3 are 1. In this particular case, without loss of generality, turns out that the 1-1-1 pattern is driving that log odds ratio relative to the null, and it's 1,000 times more than expected by chance in the data. So this is a pattern that we actually care about, and we store this information. For other groups of genes, you might actually see it's the 1-0-1 pattern that's doing this, and so on. So it doesn't need to be just expression. It could also be a combinatorial absence as well as expression that is there. So now you actually do something very simple. We start with the first cell, so we extract all these combinations that stand out relative to the null, and then we do something simple. We ask, for the first cell, does that cell have this pattern 1-1-1? If yes, we call it a 1, and if not, we call it a 0. We move on to the next pattern. Does the first cell, again, have this other pattern amongst G2, G3, G5? Yes or no? And that becomes a 1, 1, and 0. And then you repeat everything for all the other cells. So now we've ended up with a new binary matrix. Similar to the one we started with, it's every row is a cell, so that hasn't changed. But instead of every column being a gene, every column is a combinatorial pattern of gene expression, which could be, as I said, like this 1-1-1 we see here. The cell has that pattern in those genes. And then this is repeated for all patterns at different orders of interaction. This is our new object. And we keep the information of what kind of gene patterns each column is representing. Now, those of you who might have seen single-cell RNA sequencing data, people usually cluster the cells. So typically it's discrete. Now, the problem is that we all know what the problem with that is. Biology is a continuum. Cells don't just belong to different discrete groups. And even the fuzzy versions of those clusterings assume some sort of locality in expression space, that things that are close are the same object. Now, that is broadly true for cell types. It's not true for cell states. So you can imagine that I have a cut of my brain tissue, and I have neurons and astrocytes. Now, there are different cell types. So in expression space, they'll be proximal. But I could have cells within the neurons and cells within the astrocytes that are dividing. That cell division, or being part of the cell cycle, is a cell state. And that structure may be non-local because there's subsets of these that are behaving in that way. So as far as type is concerned, they are different and they're local. But as far as the state is concerned, they may be in the same biological state. They may be dying cells. They may be active cells. They may be cells that are maturing. And those are specific expression patterns that get lost if we just cluster the data. And so what we're going to end up doing is not cluster cells, but cluster patterns of expression. So we flip the problem on its head, and we end up inferring these high-order programs in a data-driven manner, and then cluster the programs. And so what you see is that a cell can appear in a branch here, but it can also appear in a branch here and a branch here. Maybe one branch represented being a neuron, another branch represented based on those genes that it's an active neuron, and another branch states that it's a mature active neuron. So now a cell, as it's more faithful to the biology, it can lie in a continuum, and it can be multiply labeled by several biological labels. So here's an example of cell cycles. I mentioned stuff in the brain. So this is the real example. On the left-hand side, we have neurons in reduced dimensional space. On the right-hand side, we have astrocytes. And again, you see that if you were to sort of naively cluster this, yes, they come in two different blobs, and they have this pattern as expected. However, so this is what I want to show you. That's the crucial bit. If I start looking at cell cycle, these genes that you see here, to a biologist, if they look at that, they immediately recognize all these genes as cell cycle. The plus and minus represents, the plus represents presence, minus represents absence. And you see that they come in combinatorial patterns. And in particular, you see that is non-local. So here in blue, we see all those cells on this plot that have this expression pattern, and that represents being in the S phase part of the cell cycle. And this structure is non-local in a sense that in expression space, they belong in different parts. And again, this is not surprising because despite these guys having two different types, they could still be dividing. And as far as cell division goes, they're in the same cell state. And that's why we see this non-local structure. It is actually rampant across the data. Here at the bottom, we were basically just trying to show that absence of a gene is as important as presence of a gene. So in fact, if you don't demand this and color these cells just by the presence, you see one big blob. But if you say it's this combination of present and this gene is absent, or and this gene is absent, as you go through the cell cycle, you really see very clear sort of density of where the cells are in this reduced dimensional space. So again, I don't want to rely too much on visualization, but what I wanted to say here to drive the point home is that clustering algorithms that rely on proximity in reduced dimension or Euclidean distances will not be able to capture this kind of pattern by construction. So we started with a very homogeneous data set. So there were so clustering would have declared this entirely as one blob. If you really pushed it with some kind of uncertainty quantification, it would maybe say, actually, maybe there are two groups here. And on the same data set, we identified 25 different states. And these states range from more stem cell-like cells to more mature cells, cells that were in the G1S part of the cell cycle and G2M, early versus late. This one's really cool. So metaphase, anaphase is the point where the cell is dividing. So we're really like catching the cell in the act as it's dividing. We saw blood contamination, activated cells, dead cells, neurons in the middle of there. So, and then these were also interesting. So cilia is sort of related to these organelles that cells use to move. And you see multiple cilia here. Turns out that some of them are non-motile, so the cell is fixed, and some of them are cilia expressing genes that allow the cell to move around. And yeah, okay, this is
overlapping sets.
They do overlap.
Yeah, they're not.
No. Yeah. So it could be that you have an IPC cell that is also in G1S that is also activated. Okay, so now this was great. So we're very sort of happy we applied it to a whole variety of cases, except that when I go and speak to my wet lab biologist colleagues, they're like, look, I don't understand your interactions and so on. I just want this plot at the end of the day, and I want my genes, and I want to be able to interpret that. So we spent quite some time doing some sort of software development on this. So this software is now in the form of a next flow pipeline. Data goes in after quality control, and what you get out is these high order interactions. And then we made a Shiny app interface where the biologists can take in those, and then as an output, they get their cells and which group they belong in. I need to emphasize that these labels are biological. So all the algorithm gives you is the genes, but at some point you have to say, okay, all these genes, combinatorial genes, are fully data-driven. And I want to emphasize that. We're not putting genes on a UMAP space. All the combinations are entirely data-driven, but there would, of course, need to be an interpretation. So biologists need to look at this and say, this is a cell cycle, or this is a motile ciliated state, and so on. So that's where the data-driven part stops. Okay. So, of course, as this is a paper that we published in 2025, and as reviewers rightly so point out, is compared to other methods. So I want to first say that STATA is the only method that simultaneously quantifies high-order expressions beyond pairwise. And this really helps it give very high resolution. It includes absence as well as presence or co-expression of genes. We have uncertainty quantification and false discovery rate control over all the reported gene modules. And the reason is we count, we bootstrap, so we have p-values, and then we can have control over FDR. So you don't need to sit there and do hyperparameter tuning and get whatever on the other side. And of course, it's fully transparent because we started with a model-independent definition in terms of probability distributions, and then we started counting. So here's a further example with other methods in the literature, such as non-negative matrix factorization and more sort of deep learning models and topic modeling. So here in bold font, what you see is anything that's bold font means STATA has more specificity in capturing those states. So some of the states were not captured by these methods at all. These are the ones that did get captured. And we see sort of the specificity is higher for these two in almost all cases. Where there's a star, again, these methods were not able to detect that kind of cell state or cell type. Now, something interesting happens here with NMFs. There are 3 cases, so minority, but still there are 3 cases in which NMFs are more specific. So now that doesn't mean that Stata hasn't found the state. It just means the gene markers used for that, there were more markers that was identified by NMF than by Stata. So in practice, it will actually not make any difference. But for the sake of comparison, I'm going to put this here. Except these methods don't have uncertainty quantification. So what happens in practice when you run this data, this is what you get. On the x-axis, we have Stata labels. On the y-axis, we have states derived by NMF. And there are states that agree between the two, actually to a large degree. The reason this is not diagonal is, again, because a cell can exist in multiple states. So that's not what you expect, but you expect an enrichment of one in the other. The problem is here, there's a whole bunch of stuff that NMF outputs that are unknown to a biologist. And probably it's because they're noise. So if there was uncertainty quantification on this, in reality, they wouldn't have actually appeared, and then we wouldn't have wasted time trying to figure out what these are. And we actually came up with a criterion on what we call known or unknown. So for Stata, we demanded 3 specific marker genes that come out as a data-driven manner. That's sort of unambiguously, biologists can point and say it's this cell type or cell state. For other methods, we were even more generous. We said, yeah, 1 or 2 gene markers is enough. Nevertheless, it was not possible to identify what's actually going on here. And on top of that, the resolution is higher for Stata. So when I talked about ciliated genes, the fact that some were motile cells, so cells that move and some that don't, this wasn't captured by any of these other methods. So then there's always the question of reproducibility. How reproducible is it? Yes. Okay, sorry, yes, I'm almost there. So there's the question of reproducibility. So here we are looking at a data set where we take the cells, again, from the brain, we split it into 2 disjoint sets, we ran Stata on one, and we sort of project it onto the other half that is not seen, and then vice versa. And what you want to see for reproducibility is that the pattern is the same. So again, don't expect a diagonal because of this multiple existence, but what you should look for is that this pattern of reds and whites is actually identical between the 2. And then we were given the harder task that, okay, well, this is from 1 mouse. What if there are 2 different mice in different labs? And can you again demonstrate in the same biological condition that this is reproducible? So we did that. And then on top of that, the reviewers started getting into multimodality and asking the question, the heterogeneity we claim is actually an order of magnitude higher than what the current methods are reporting. So if this heterogeneity is real, do you see it in a different data modality, such as chromatins being open or closed? And again, we demonstrated that the heterogeneity exists in other data modalities too. So beyond normal biology, we applied Stata to disease data. So here we're looking at a liver that's uninjured versus cirrhotic diseased liver. Again, the x-axis are the cell states, and then what we see here in bright red are those states that are enriched in disease versus uninjured. So perhaps any state that's common between the 2, we don't really care about, but we actually care about those states that we see appear in disease. And again, if you zoom in on some of these states, we ended up reporting multiple subtypes of cells in a tissue that were missed otherwise in the disease case. And again, this has also been applied to cancer data to associate cell states with survival in patients. And yeah, long story short, this whole thing is available as an app, for sort of visual app for biologists to engage with. And yeah, if you happen to work in this area, we'd be more than happy to help anyone to apply this to data. That's all. Thanks.
So for estimating higher modes or interactions between multiple genes for high dimension, how many samples do you need? Doesn't the sample complexity grow exponentially?
Yeah, so that's the beauty. Because you are inferring this Markov blanket, so this graph that was becoming sparser and sparser, you actually end up for every given gene, maybe you need 10 or 20 other genes. So it's independent of the rest of the genes. So that reduces the dimension in a way. And if you're only looking at a reduced dimension, so this was done even 5,000 cells, 10,000 cells, 50,000 cells. And so in each of them, we end up getting the interactions out.
the uncertainty in those interactions small, but it's finite and it might be.
Yeah, so as I said, so we haven't really managed to, with 50,000 cells at the largest, we haven't managed to go beyond 7-point interactions, because as you go from 2 to 3 to 4, the error bar grows. But getting 3, 4, 5, yeah, that's fine.
I guess this kind of sounds like that, like each gene module, like, is there equivalent to like something like a reaction pathway, reaction database, or if you substitute it rather than using the causal discovery methods, was there like equivalent?
So just to answer your first question, we binarize at the beginning because we don't want to fit, basically, so we count. When we start annotating the cells, so at this point where we start looking at these branches, we go back to the original expression space, because typically the way to annotate them biologically is by comparing differential expression. Differential expression methods rely on sort of, say, negative binomial distribution and so on. And we also don't want to be restricted to just the 1,000 variable genes. We want everything. So there we use the unbinarized version. So binarized at the beginning and then unbinarized at the end. First question. Second one, yeah, reactome. So we compared a lot at the beginning when we were trying to see if our graph is actually causal. The problem with a lot of those reactome pathways is that if you look into it like a string database, they're not also always causal. Sometimes a lot of them are correlational, as we see here. I have not tried replacing the whole thing with what's in there, because it's also, there's a lot of tissue-dependent context as well. So, you know, reactome could be a general expression, but then we're dealing with a specific tissue in a specific disease model in a specific situation. So we want to sort of derive it from the ground up.
Quick question. So sometimes with gene expression, it doesn't capture transcription factor activation because it might be through a signaling pathway, like a phosphorylation event. So in that case, you've basically got like hidden variable, like your gene expression data isn't like sufficient to capture these relationships. And there's some hidden variables like phosphorylation or, you know, estrogen receptor or something like that. And that switches on loads of genes. So in that case, is this kind of looking for looking for sparser kind of groups going to struggle if there's a broad hidden regulator that sort of activates like 1,000 genes? Because then you have to discover all the pairwise, like n squared of all those activated genes or something.
Yeah, so I think there are several things I can say about transcription factors. You are completely right in saying that sometimes it turns on and then it just turns off for a long time and it has already activated a huge amount of stuff. So one thing we end up doing, if people know the transcription factors involved, we try to sort of include it as part of the model. But then again, it may not show a large dependence from RNA expression data because it's just turned off very quickly. The result of that, as long as it's stochastic downstream, so they're not always one throughout the data, or like one, they're in parts of the cells, but not other cells, you will still be able to capture the difference as far as cell state goes.
Yeah, I guess my question is more like, if you have a hidden variable, it means the graph,
yeah,
the relationships can become very high dimensional. Like,
yeah, this is
you're a
true.
ferromagnet. Like,
This is
example,
true.
you've got these
Yeah.
neighborhoods of hidden variables,
Yeah.
you just deal with a
Yeah.
clique in the graph.
Yeah.
or something.
Yeah. So that can be, I guess, an issue, because a lot of these also algorithms that do causal discovery, if you like, that they are assuming that you don't have unobserved confounders. So if you have that, then obviously that's going to mess things up. The only thing I can say is that for now, relative to what we're doing, we're getting a higher resolution. But that's all I can. Yeah, that obviously you haven't solved everything. So yeah, that's yeah.
All right.
Thank you.